Bismillahirrahmanirrahim. Assalamu alaikum, dear student. I am Professor Dr. Shahzad. Today we are going to discuss endocrinology. What is endocrinology? It is the intracellular chemical communication. What are endocrine system for? Endocrine functions to maintain internal homeostasis, to support cell growth, to coordinate development, to coordinate reproduction, and to facilitate responses to external stimuli. This is the structure of hypothalamus and pituitary gland. Almost all secretions of pituitary gland is controlled by either hormonal or nervous signals from hypothalamus. Secretions from posterior pituitary is controlled by nerve signal that originate in the hypothalamus and terminate in posterior pituitary. In contrast, secretions by anterior pituitary is controlled by hormone called hypothalamic releasing and inhibitory hormones or factors. They are secreted by the hypothalamus and pass to the anterior pituitary through hypothalamic hypophyseal portal system. The release and in some cases production of each of the pituitary hormones are under tonic control by at least one hypothalamic hormone. The hypothalamic hormones are released from hypothalamic nerve fiber ending around the capillaries of the hypothalamic hypophyseal system in the pituitary stalk. They reach the anterior lobe through the special portal system that connect the hypothalamus and anterior lobe. The hypothalamic hormones are released in a pulsatile manner. The anterior pituitary target cell respond better to pulsatile administration of these hormones than to continuous exposure. Corticotrophin releasing hormone having 41 amino acids stimulate corticotroph cells of anterior pituitary to secrete ACTH mainly. This corticotrophin releasing hormone has secondary or lesser effect on lipotrophin and melanocyte stimulating hormone or and endorphin. Target gland hormone affected by this is hydrocortisone. The release of ACTH is primarily controlled by corticotrophin releasing hormone or CRH, but a number of other hormones including antidiuretic hormone, catecholamines, VIP, and angiotensin II may be involved. Corticotrophin releasing hormone release is influenced by cortisol, a glucocorticoid hormone which is secreted by the adrenal gland. This thyrotrophin releasing hormone, which is a tripeptide having three amino acids, stimulate thyrotroph cells of the anterior pituitary to secrete thyroid stimulating hormone or TSH. The target gland hormone affected are T3 and T4, which are also called thyroid hormones. T3 is also called triiodothyronine and T4 is also called thyroxine or tetraiodothyronine. The TSH release The TSH release is primarily affected by TRH or thyrotrophin releasing hormone, which in turn is regulated by the thyroid hormones, which are T3 and T4, but TSH release is also inhibited by somatostatin, which is a growth hormone release inhibiting hormone. The gonadotropin releasing hormone having 10 amino acids stimulate gonadotroph cells of anterior pituitary to secrete LH and FSH. LH is a luteinizing hormone and FSH is follicle stimulating hormone. Target gland hormones affected are androgens, estrogens, and progesterone. The release of LH and FSH is controlled by concentration of one releasing hormone, which is called gonadotropin releasing hormone. This in turn is primarily regulated by circulating levels of gonadal hormones that reach the hypothalamus. Then growth hormone releasing hormone. Growth hormone releasing hormones having 44 amino acids stimulate somatotroph cells of the anterior pituitary to secrete growth hormone. Target gland hormones affected is IGF-1. Growth hormone releasing hormone stimulate the rate of transcription of growth hormone gene. Growth hormone release and productions are under tonic control by both stimulating and inhibiting hypothalamic hormones. In addition, peripheral feedback loop is involved in growth hormone regulation. IGF-1, also called somatomedin C, which mediates some of the effect of the growth hormone, stimulate the release of somatostatin, also called growth hormone release inhibiting hormone, while inhibits the release of growth hormone releasing hormone. Growth hormone release inhibiting hormones having 14 amino acids stimulate somatotroph cells to inhibit secretion of growth hormone. Target gland hormone is IGF-1, T3 and T4. somatostatin or somatotrophin release inhibiting hormone are other names of growth hormone release inhibiting hormone. The regulation of prolactin regulation of prolactin biosynthesis and secretion by the lactotrope cells is primarily under the tonic inhibition by prolactin, sorry, the regulation of prolactin synthesis and secretion is primarily under tonic inhibition by hypothalamic agents. It is unique because of combined neural, that is nipple stimulation, and neurotransmitter neurohormone link. Prolactin release inhibiting hormone inhibit prolactin secretions by lactotropes. Dopamine inhibits the prolactin biosynthesis by inhibiting transcription of the prolactin gene and release, but does not account for overall prolactin inhibition. Thyrotrophin releasing hormone stimulates the rate of transcription of prolactin gene, as we have seen while we were discussing thyrotrophin releasing hormone. Many of the hypothalamic hormones, in particularly thyrotrophin releasing hormone, corticotrophin releasing hormone, and somatostatins, are found in other portions of the nervous system and in variety of peripheral tissue. Gonadotrophin gonadotrophin releasing hormone associated peptide, that is GAP, is a 56 amino acid neuropeptide, has both gonadotrophin releasing hormone activity and prolactin prolactin release inhibiting hormone activity. Gonadotrophin releasing hormone activity and prolactin release inhibiting hormone activities. This gap is a potent inhibitor of prolactin release and may explain the link between gonadotrophin releasing hormone and prolactin secretions. This all is about today's lecture. Thank you very much, dear students. Allah Hafiz.